Okay, welcome to NEET World again. This is the third demo class, I think. Fourth demo class, yes, thank you. Today we are going to discuss about this locomotion and movement. So before that, welcome to my channel. So these are the demo classes for the NEET long term 2027, right? If you want to join this NEET long term 2027, please contact the below numbers, right? Thank you. So I think, am I audible, right, online? Right, am I audible? Thank you. So today we are going to discuss a new chapter, that is locomotion and movement. So this locomotion and movement consists, from this thing you will get around 3 questions, right? So this is very sure questions. You will definitely get minimum of 2 and maximum of 3 questions you will definitely get. So in this entire locomotion and movement chapter, what are the things you are going to listen? So the first thing is, what are the types of movements? After that, coming to the human muscular system, how the muscle is made up of, how the structural and functional unit of life is, what is that? And after that, coming to the skeleton system. In skeleton system, only this joint part is there. After that, overall disorders of both muscular system and skeletal system. Definitely you will get one question from here, and you will get maybe from muscle or joints, you will get one question from here, and overall they will combine all these things and they will give one question, which means they will not give one question from muscle, they will combine the muscle part and also this skeleton part and also this joint part, and they will combine all the options and they will give one question. That's how nowadays they are framing the questions, right? So we have to study each and everything from this unit, then only you will able to do definitely 3 questions perfectly, right? For that we will study entire thing. In this class we will study only the types of movement and muscle, right? So these types of movement is the basic thing to understand. With the understanding of the types of movement only we can understand the muscle and all these things, right? So let's start locomotion and movement. The first question we have to know is, what is locomotion and what is movement? So to think, this locomotion and movement are looking same, but they are not actual same. So what is locomotion, what is movement, we have to understand very clearly. So let's take me example. I am talking and I am moving, I am doing so many things, right? So these are all consist of locomotion and movement also. What actual locomotion, what actually differentiate between these two things? For example, let's take my hand. So the structure of my hand is looking like this thing. Now I am doing something and what happens? The structure was changed, which means movement is nothing but changing in the structure of anything, that is called as movement. For example, coming to locomotion, in locomotion not only the changing in the structure of the thing, it leads to the change in the position of an animal. For example, I was moving from here to here. What happens? My legs are moved. Simultaneously what happens? My exact location, I am here right now, after few minutes I was here. So what happens? My location was changed. So in locomotion what happens? The location, or simply you can remember, position of animal with respect to place, right? It is going to change. That is called what? Locomotion. Whereas in movement there is no change. There may be or may not be, but in locomotion particularly there will be a change in the position of animal, whereas in movement there will be no change, there will be only change in the body forms. That is called movements and this is called locomotion. These two are looking like interconnected, right? So that's actually true. For example, if you see the NCERT statements, what they are telling, locomotion is the voluntary movements resulting in the change in a location. So locomotion directly, the aim is a change in a location of an animal. So it is called what? All voluntary movements. So I am making movements which I want to do. I am making these movements, I want to do. So these are all called voluntary movements. These movements are collectively called this locomotion. These movements are collectively and doing what? Changing in my position. So this is called what? Locomotion. And these small things are called as what? Movements. If you see this statement, all locomotion are movements, but all movements are not locomotions. What it is? For example, there are some movements was happening in my body. For example, I was here, I was doing lot of movements. I was talking, my eyebrows are doing something, and I also, my fingers are doing something, and my lips are doing talking. So these are all called what? Movements, right? Whereas with all movements, what I am doing? I am changing my position. So which means all locomotion, for to do a one locomotion for an animal, there will be definitely what happens? Movement has to take place. With no movements, there will be no locomotion, which means all locomotions are movements, but all movements are not locomotion. So here I am doing many movements, but I am moving, I am not moving, I am not doing any locomotion, just I am doing movements only. So all movements are not locomotions, but all locomotions are movements. All locomotions done by only movements. Understand? So this sentence, all locomotions are movements, right? So for locomotion, there will be movement has to take place, but all movements are not locomotions. Maybe that movement cannot be perform locomotion, can be perform locomotion, but not all movements are not locomotion. Understand this statement? They will give they will give definitely this question. So same kind we have in non-coordinates and coordinates. Likewise, there are few statements. These are very important. Understand what is locomotion and what is movement. There is an actual difference with respect to change in position of an animal. That is the only thing. And I said these two are interlinked. How they are interlinked? Let's see. So there is an animal called paramecium. So they have some structures called what? Cilia. So with the help of cilia, the paramecium is going to change its position. So it is moving with the help of what? Cilia. And also this cilia is not performing only locomotion, it also performs what? Food capture. So what happens? For example, let's take, so this is paramecium and all these are what? Cilia. So these cilia are doing some movements. With the help of movement only, it is moving. So this is called locomotion. That's how this movement and locomotion are interlinked. Without movements, there will be locomotion. There will be not locomotion. So this cilia is performing some movements. So with the help of this movement, what happens? This this animal is performing locomotion. And very simple example, we know sperm. We know, right? Sperm. For example, this is flagella. So this is flagella. Flagella is showing some whip-like movement. So that is one movement. So with the help of flagella, with the help of the movement of this tail, what happens? This entire sperm is moving from these positions. So this how this movement and locomotion are interlinked. So when we think this locomotion is only for this changing the position of animal, that's not true. Not only for the changing of the position of animal, it is also for food capture. There is a best example called paramecium. The cilia is helpful in the food capture also and also performs locomotion. And also there is another example called hydra. They have tentacles to capture prey and also for locomotion. So this thing you will understand in animal diversity. And also we have limbs. We have limbs, we can do anything. We are changing our body postures such as called movements, and with the help of movements also we are doing locomotion. So these are the few examples to know how the movements are interlinked with locomotion, right? So this is locomotion and movement. And we'll see what are the types of movements that are there in our human body. There are three different kinds of movements. One is amoeboid movement, another one is ciliary movement, another one is muscular movement. These things you know in previous. Let's see revise. So the first one is amoeboid movement. In animal tissues, in previous class, in animal tissues we already discussed these all movements, right? So the first one is amoeboid movement. There are some cells called as macrophages. There are some cells called what? Macrophages. These macrophages, what is the function of macrophage? Anyone can? What is the function of macrophage? Yes, engulfing the bacteria, right? So there is, with the help of this pseudopodia, so macrophage what is doing? It is taking the food, right? So for example, this is one macrophage. If you see, so it has nucleus. So with the help of this pseudopodium, what happens? It is capturing the prey, whatever it is, food or it may be pathogen. So it is capturing what? That prey with the help of what? Pseudopodia. This kind of movement, this pseudopodial movement, this kind of movement is called as what? Amoeboid movement, because this pseudopodia is exclusively we can see in amoeba. So that's why we gave one name which is called as what? Amoeboid movement. So we have macrophages which showing amoeboid movement, right? You can see macrophages and leukocytes, monocytes. So yesterday we already discussed, for example, this is blood vessel. So here what are there? Monocytes are there. So these monocytes are doing what? Circulating in bloodstream. So whenever there is a tissue and also there is a pathogen present in tissue, what happens? This monocyte is entered into that particular tissue. Then what happens? This same monocyte is becoming what? Macrophage. Right? Yesterday we discussed, right? These are tissue fixed macrophages, whereas these are wandering macrophages. Right? The same cell, here it is called as what? Monocyte. The same cell, it is called as monocyte. Whenever it is entered into tissue, then it is called as what? Macrophages. How it is entering? With the help of this amoeboid movement. And the process of entering this monocyte into the particular tissue is called as diapedesis. With the amoeboid movement, it will enter into the particular tissue, which means our body has some cells which performing some kind of movement which are similar to amoeba. So that's why we gave one name which is called as amoeboid movement, right? And coming to next one is ciliary movement. In yesterday class we discussed ciliated columnar epithelium cells, right? So there are some cells, for example, this is one duct, fallopian duct. So in this fallopian duct, these are kind of ciliary kind of epithelium is there. What it is doing? It will movement this egg from one direction to in that particular direction. What happens in our body? There are some cells which are called as modified cells of epithelium. So these are called as ciliated epithelial cells or ciliated columnar epithelial cells, which are doing what function? Locomotion. Which means, so there are, we already discussed two examples. One example says it is present in fallopian tube of female reproductive system. Another example is inner lining of trachea or bronchioles. What happens in that? So these kind of cells are present, they are having cilia. They allow the movement of molecules in one direction, right? From ampullary region to, it will capture the egg in ampullary region. From ampullary region to, it will send the egg into the uterus, right? That's how there are some movements was happening in our body. Such movements are called as what? Ciliary movements. Understand what is ciliary movements? And next type of movement is muscular movement. It is exclusively made up of what? Muscles. So that movement with the help of these muscles is called as what? Muscular movement. We will see what actual muscular movements are there. Apart from this amoeboid movement, ciliary movement, muscular movement, there is another type of movement. We already discussed this thing, which is called as flagellary movement. So we have sperms. Whenever the sperm has to travel, it has what? Tail. it is showing what? Flagellary movement. So because of that flagellary movement, this sperm is entering into the egg, right? So that's how in our body we have different kind of movements. First is what? Amoeboid movement we discussed. Second one is what? Ciliary movement. Third one is muscular movement, and fourth one is flagellary movement. Understand? So these are the types of movements with examples. So the next one, we have to know what are the different types of muscles we have. Actually we discussed in these types of muscles in tissues. Again we will revise. So based on the some functions and locations, muscular types, muscle structure, we have classified the muscle into three different kind of thing. One is what? Skeleton muscle. Second one is what? Smooth muscle. And third one is what? Cardiac muscle, right? So these three things we yesterday discussed. Let's see the structures and let's see the properties of all these things comparatively. For example, if you see the structure, here it is cylindrical, straight long, but here it is not cylindrical, it is fusiform shape. It is fusiform shape, tapering at both ends. It is also tip. This end is also like tip, and this end is like tip. So tapering at both ends, there is a word called. So this is fusiform shape. Whenever if you see, it is long and cylindrical, but is unbranched. But if you see, it is also long and cylindrical, but here it is showing what? Branching pattern. So that was the structural difference between. So it is having straight, long, cylindrical, unbranched. It is also straight, long, cylindrical, branch. It is exactly opposite. It is just fusiform shape. If you see, this is entire one cell. It is the one cell having multiple nucleus. But if you see, it is only one nucleus. These two are multinucleate, whereas it is uninucleate. Understand the structural difference? See, these skeletal muscles are attached to, these muscles are attached to skeletons, and we know one thing: the muscle or the thing which is attached to bone to muscle is called as what? Tendons, right? These are the thing which helps to bone to muscle, and there are something which is called as ligaments. This helps bone to bone attachment. So in skeletal muscles we have what? Tendons. And next one, if you see, there is one point which is called as striations. Striations are nothing but, if you see, there is one light band and there is one dark band. There is one light band, there is one dark band. That's how the muscle was framed. So that's showing called what? Striations, right? That is called striations. And it is showing what? Voluntary nature. We can control this skeleton muscles. Whenever we want to take anything, we can control all these muscles. So that's why it's called voluntary. And it has rich blood supply, and because of the rich blood supply, it will cause fatigue condition. So these are called fatigue muscles also. And you can see muscle fiber is multinucleate. Because why? So this entire cell is having what? Many nucleuses. Yesterday I already told you why this multinucleate condition is coming. Why this multinucleate condition is coming? So whenever there is a cell, it will divide. So there is one nucleus. Whenever it is involved in cell division, what happens? First of all, nuclear division will happen. Next, what happens? This cell division will happen. In some cells, what happens? First of all, nucleus division will happen, but there will be no cell division. So karyokinesis is happening, but there is no cytokinesis. So without cytokinesis, what happens? This is looking like single cell and having two nucleus, right? This is single cell having two nucleus. The same nucleus is going to divide another time. So the same nucleus is going to divide another time. So that's how one single cell having what? Multinucleate condition. This condition is called as what? Syncytium condition. Understand? That's how the multinucleate condition was came. But coming to here, it is what? Uninucleate. It is having low blood supply. That's why it is non-fatigue. These are involuntary in nature. We cannot control these things. So that's why it is involuntary in nature. And here if you see, there is no light band, dark bands. Why light band, dark bands? We will see now. So here there is no light bands and dark bands. So there will be no striations. And all these smooth muscles are present in where? Smooth visceral organs. right? So comparatively, if you see here, this cardiac muscle exclusively present in heart wall and is also showing light bands and dark bands, that's why it's called as striations, and these are also involuntary in nature. We cannot control our heart. It is why it is myogenic heart, right? In circulations we will discuss heart is myogenic, and it is also rich blood supply, and here there is a unique point, it is non-fatigue, and also it is uninucleate and more mitochondria. So in between this all branches, yesterday we discussed there is one junction which is called as what? Related disc. So in epithelial tissues we discuss three different types of junctions. One is what? Tight junctions, another one is anchoring junctions, third one is gap junctions. These tight junctions are responsible to prevent the body fluids, leakage of body fluids. The first junction we will discuss again, no problem. Tight junctions function is to prevent the leakage of body fluids. And coming to this anchoring junctions, the function of anchoring junction is to hold the cells together, to anchor the cells together. And the third one is gap junction. What is the function of gap junction? The communication. To communication between two cells, there are some junctions which is helpful for the communication. Those are called as what? Gap junction. Same like here we have called intercalated discs. These intercalated discs are responsible for what? Communication. It's like helping like gap junction. Sir, what communication was happening here? See, whenever it is atria, here we have what? Sinoatrial node. Whenever impulse is generated, what happens? This entire cell has to initiate, right? So first of all, what happens? This cell will be initiated. So generally this cell will be initiated. After in a neuron, if you see, what happens? Depolarization. First this portion will be initiated, after this transmission will occur like this, after this like this. So it will take some time, but with the help of this intercalated disc, what happens? This simultaneously all the cells are going to contract at a time. Why? Because intercalated discs are present. This allows the impulse from one cell to another cell simultaneously. So likewise, whenever this cell is initiated, same time this cell also will be initiated, this cell also will be initiated. That's how intercalated discs are very useful in cardiac muscle. So they will give, intercalated discs are responsible for these functions. They will give to prevent the body fluids, to hold cells together, for communication. The answer is what? Communication. To communication, these intercalated discs. So these intercalated discs are present in between two cells or in between two branches in the wall of heart, right? So that was the three different types of muscles which are present in our body. One is what? Skeletal muscle, smooth muscle, and heart muscle, right? So after, now we need to study brief on skeletal muscle, how it is looking like, how the skeletal muscle is made up of, all these things, right? We will study. So before that, there are few points. If you see, human muscular system is entirely made up of muscles which are mesodermal origin. Yesterday we discussed in epithelial tissues, muscle is from mesodermal origin, connective tissue is from mesodermal origin, epithelial tissue is from ectoderm, mesoderm, endodermal origin. So that thing you need to remember. Likewise, this muscle is also from mesodermal origin. This muscle constitutes 40 to 50% of the body weight. So that much muscles we have, right? So this muscles showing few properties. What are the few properties? The first property is excitability, and the second property is contractility, and third property is extensibility, and fourth property is elasticity. These things we discussed in epithelial tissue. Let me revise. So what is excitability, right? So for example, let's take, this is one muscle. This is one muscle. So muscle has to contract, relax. That is the function of the muscle, right? So whenever the signal from the neuron it is coming, what happens? It has to receive or it has to respond to the particular signal. That is called as what? Excitability. Then what happens? Whenever it is receive or respond to the particular signal, what happens? It will initiate the muscle contraction process. So that response, receiving and initiation, this everything is called as what? Excitability. Likewise, neuron also having this excitability property. Next, contractility. So what happens with the help of this receiving signal, the ultimate goal of the muscle is what? Contraction. So shortening of this muscle fiber. So this is called as contractility, that property of a muscle, showing shortening of this muscle fiber is called as what? Contraction. Extensibility is nothing but extend, elongation of muscle fiber. So previous the muscle is this thing, so whenever I am doing something, this muscle stretch is going to extend. This stretch or increase in the length of this muscle fiber is called as what? Extensibility. Decrease in the length of the muscle fiber is called as contractility. And after few time, what happens when I releasing some force, what happens, this muscle, whenever I stretch or whenever I contracted the muscle, it is coming back to its original position, right? After stretching the band, after removing the force, what happens, the band is coming to its original position. That thing is called as what? Elasticity. This muscle is showing that property. After contraction, what happens? It's coming back to its original position. That property is called as what? Elasticity. So these are the four properties which were shown by what? Muscle. Understand the definitions of all these things, right? Now we will see the structure of actual skeletal muscle, how it's look like. See, this is what? Bone. This is what? Bone. From bone, this muscle was coming. So from bone to muscle, there is one thing we know, right? That one is called as what? Tendon. Tendon is the one which attaches this bone to muscle. So from bone, we have this entire muscle, right? We have this entire muscle. This entire muscle having different kind of cells or different kind of groups. We will see. So this entire muscle is made up of some round or sac-like structures. All these structures are called as what? Fascicles. All these structures are called what? Fascicles. Inside these fascicles, there are some round color structures. I am drawing with black color. Just see. So these are called as what? Muscle fibers. Right? There is a big thing called muscle. In that big thing, there are small muscle fibers are there. In small muscle fibers, I am drawing again some dots. These are called muscle cell or myofibril. Understand? So for example, this is entire muscle. This entire muscle is made up of what? A group of fascicles. So entire muscle fiber is made up of fascicles. In each fascicle, it is having muscle cell. In each muscle cell, we have what? Myofibrils. In myofibrils, what are there? In myofibrils, this actin and myosin will be there. Understand? So how the organization will be there? So I will draw. Sorry. Right. So if you see, this is one entire muscle bundle. This bundle is made up of a group of small groups which are called as what? Each one is called as what? Fascicle. See, they mention what? Fascicle. In each fascicle also, there are few groups of cells, and each group of cell is called muscle fiber. Muscle fiber or muscle cell. And each muscle cell also, there are some dots, right? So what are these things? So for example, I elaborate this muscle fiber. Each muscle fiber also having what? Myofibrils. This is the organization. Sometimes they will give, arrange the following cells in the sequence. The first one will come what? Muscle bundle. In muscle bundle, what is there? Fascicles. In fascicle, what is there? Fiber, or called as muscle cell. In muscle cell, what is there? Myofibril. Myofibril. In myofibril, what is there? Actin and myosin. This is the structural organization of one muscle. Understand? And here, what are they? in each and every cell there will be a plasma membrane. We have a names for each and every cell. For example, there is one plasma membrane which surrounds the entire muscle bundle, that is called as what? Epimysium. Epi means top layer. So the top layer which is surrounding the muscle bundle is called as epimysium. So there is another layer, so that is surrounding this fascicle. That fascicle is called as what? Perimysium. If it is inside, then what is called? Endomysium. Likewise, if you see that fiber cell, endomysium, right? So epimysium is covering the top muscle bundle. Inside we have what is called fascicles. Fascicles was covered by perimysium, and inside muscle fiber is there or muscle cell is there. This muscle cell is covered by what? All these muscle cells are covered by what? This endomysium. And inside myofibrils, and all these myofibrils are covered by sarcolemma, which means this myofibrils are covered by sarcolemma. I will draw very neat. First one, muscle bundle, after that fascicle, after that what? Muscle fiber, or also called as cell, after that myofibril. Here we have actin and myosin. The starting thing which is called as epimysium. After that the fascicle is covered by what? Perimysium. After that this myofibril or cell, it is covered by endomysium, and this myofibril is covered by sarcolemma. This sarcolemma is looking like what? This thing. This is sarcolemma. Inside we have what? Myofibrils. If you elaborate this myofibril, it's looking like this and having what? All actins and myosins. It is light band, it is dark band, and actins inside myosin. So these are all what? Light bands and dark bands. So this will constitute all this myofibril, and all myofibril will present in cell, all myocells are present in fascicle, all fascicles are present in muscle bundle, and this muscle bundle also looking like what? Striations. That's how dark and light bands will come. Understand the organization and layers of all these things, right? Epimysium done, perimysium, fascicle. This entire thing is fascicle, and inside muscle fibers are there. Inside muscle fibers, myofibril is there, right? Next one. If you elaborate this muscle fibril, for example, if you see this is muscle fiber is surrounded by what? Sarcolemma. So this myofibril, see this is one myofibril, these are myofibrils. See, all myofibrils are surrounded by sarcolemma. All muscle fibers are surrounded by endomysium. That's how. For example, this is one myofibril. In myofibril, there are some light bands, dark bands, light bands, dark bands. One I band, another I band, from one Z line to another Z line. Here we call it as what? Sarcomere. We will see how the sarcomere will look like with help of some diagram, right? Right. Red color, black. This is one Z line. Sorry. Right. This is one actin, this is another actin, this is one actin, this is another actin. Right. Sorry. Next red color. So this red color representing what? Myosin. This black color representing what? Actin. Let's see. I am drawing another line here. This is called as what? M line. These are what? Z line. This is also called Z line. Understand what is Z line? It is having actin, and this Z line is also having actin. This M line is having what? Myosin. See, this red portion is called as myosin. Here we have some bands or zones which are called as I band or A band or H zone, M zone, like kind of thing. So what is I band? What is A band? I band is called as isotropic band, whereas A band is called as what? Anisotropic band. Sir, what is isotropic, anisotropic? If you see here in this portion, particular portion, whenever the light it will reach to this position or it will reach this muscle, what happens? It will follow the same refractive index. It is all same. So that's why this portion from here to here, this portion is called as what? I band. Right? For example, if you take this portion, what happens? First of all, it will reflect on myosin, and it will reach to actin, and it will reach to myosin, and it will reach to actin, myosin. So it is having dissimilar, it is not having same. So that's why this little portion is called as what? A band, an isotropic band. Whereas isotropic band having what? Similar kind of refractive index, whereas here A band consists of what? Different type of refractive index. Understand, it is very important. This is A band, this is this is I band, this is A band, and here this thing is also what? I band. Here we are discussing about sarcomere. The sarcomere is the present or the portion in between two Z lines. This portion is called as what? Sarcomere. Right? For example, if you see this entire zone is called as Z or I band. So here only we are representing half of the portion. So half of the I band, another half of the I band, another full A band is called as what? One sarcomere. Understand? So the structures of this A band, still we are at starting position only, right? If you see, this dashed line is called as what? M line, and this zigzag portion is called as what? Z line, right? Now if you see this area, for example this red color portion, and having what? Myosin, actin, myosin, actin, myosin, actin, all this portion is called as what? A band. If you see here this portion, it is only having what? Myosin portion only. This portion is having what? Only myosin. There is no actin, only myosin. That region is called as what? H zone or H band or H zone, Hensel's zone. If you see this portion, it is having what? Myosin, actin, myosin, actin, myosin, actin. This portion is called as what? O band, which is called as overlapping zone. Overlapping nothing but it is overlapped with myosin, actin, myosin, actin, myosin, actin, myosin, actin. Right? So this portion is called as what? Overlapping zone, and this only the myosin portion is called as what? H zone, and only actin portion is called as what? I band. That's how the different things are present. This is I portion, and this is O, and this is H. Likewise here also it is O and it is I. So these are the different areas that are present in one sarcomere. Understand? Now what is very important? Whenever contraction happens, what it is? Whenever contraction happens, what happens? That is the very important. Whenever contraction happens, what happens? There is a say called these two Z lines are coming to each other, which means they are becoming shortening. We know the muscle contraction happens where? Whenever muscle activation, whenever muscle contraction happens, whenever this thing, two Z lines comes near to each other. Near to each other. So this Z line will come and this Z line also will come. Now what happens? We'll see. Whenever the Z lines are coming, what happens? this will come like this and this will also come like this, right? So there will be an increase of O band and there will be a decrease of H band. There will be no only myosin portion. Now what happens? All this portion is covered by this actin myosin, actin myosin, actin myosin. What happens whenever the contraction happens? This H will be decreased, H zone will be decreased and O will be increased. And also what happens? This actin portion also will be increased. Right, I band will be decreased because, so this portion, this Z line also coming near to this thing, this Z line also coming near to this thing, so definitely I band will be also decreased. These three things you have to remember. I band definitely decrease and also H zone definitely decrease. Only one portion is getting increased, that is overlapping zone. That is the only one is getting increased. Understand? So overlapping. So initially this 50% was only overlapped. After contraction, this entire thing was overlapped. Initially this 50% having only myosin, now there is no 50%, no 0%, only 0% is only having myosin. So that's why H zone will be decreased. And the only thing which is remains the same is what? A band. The only thing, A band is remains same. These are the few things you have to remember for the competitive purpose. They will ask only this thing. For example, if you see what A band is remaining same. For example, if you see what I told, A band from this myosin portion to another myosin portion, that is called as what? A band. Here myosins are not getting contracted, only these Z lines are coming to each other. So this portion, it is not any effect in this contraction. So definitely this will be same. This will be same. That's why A band is not having any involvement in increasing or decreasing. It will be same. What they are doing, only I band is losing its and H zone also losing and O is only getting increasing its strength. So this will be happen in muscle contraction. Understand the muscle contraction, the structure of this muscle fibril and sarcomere, and whenever the contraction happens, these are the things which are going to done. So we have numbers in our channel. In shorts we have numbers. If you call to the numbers, they will tell all the phase details and they will send some links to you to verify that, right? So this is called muscle fiber and all the structure. Understand? I hope it is clear. Right. Next one, in this muscle fibril, what are the things that are present? Actin and myosin. That thing also we have to discuss. We have to know about the structure of actin and structure of myosin. There are some group of globular proteins. These are called as what? G proteins. G is representing what? Globular proteins. G proteins. Now what happens? These all G proteins are connected to each other in a helical way. I will draw that. So in a helical way, like this thing. So this is called as what? One filament kind of thing. So that's why we gave name, this is called as one F-actin, fiber. This is one looking like one fiber. So this is monomer globular protein. So monomer thing is called as G protein. It is not monomer, it is a polymer. Many monomers are coming together and having what? Polymer. So this is what? F-actin. So this is G-actin, that is F-actin. Understand? Likewise, it is not having only one F-actin, it is having what? Two F-actins. See. Likewise, two F-actins. This is one F-actin, this is another F-actin. two F actin polymers constitute what? One actin. This is one actin. So in this there is one line, if you draw like this, that line, that is one protein. That protein is called as what? Tropomyosin. So these are what? F actins, and this line is called as what? Tropomyosin. Here if you see, it was crossed. To hold this two F actin and tropomyosin, we have some proteins. What are the proteins? There are three different types of proteins. All together what? Troponin. There is 1, 2, 3. All together we can call them as troponin. So 1, 2, 3. So we'll give troponin I, troponin T, troponin C. So one is I, one is T, one is C. They have some functions. What are they? We'll see. Troponin C for the responsible for the calcium binding. Whenever there is a release in calcium in muscle, what happens? It will go and bind with troponin C. Troponin T, it is attached to tropomyosin. It will attach to tropomyosin. Whenever there is another thing which is called as troponin I. I is responsible for inhibition. Inhibition of what? I will draw. So this is one actin. This actin has one site which is called myosin binding site. Actins have what? Myosin binding site. Shortly, these are called as what? Active. Just remember, active site. Right? Active sites. So likewise, all these things are what? Active sites of all these things. Myosin binding sites. Right? On these myosin binding sites, what are there? Tropomyosin is there. Again black. All these myosin binding sites are covered by what? Tropomyosin. Understand the structural feature? All these myosin binding sites are covered by what? Tropomyosin. This will be done by this troponin I. It will inhibit this myosin binding sites. It will inhibit the myosin binding. It will not expose. So what happens? Whenever there is an impulse, or in your board, whenever we want to contract, or whenever the neuron is sending signal, what happens? We'll see. There is an cell, here I will draw. This is the outer layer, sarcolemma. So all the impulses traveling and coming to the sarcolemma. What happens? Impulse was generated inside. Now there is a release of calcium. From where? Endoplasmic reticulum. Just remember, calcium is releasing from endoplasmic reticulum or sarcoplasmic reticulum, because we are dealing with the muscle, right? That's why it is called as sarcoplasmic. Just remember, endoplasmic reticulum, sarcoplasmic reticulum. So now what happens? This entire cell is filled with calcium. So calcium will come and bind with what? Troponin C. It's like story. Calcium will come and bind with what? Troponin C. Whenever calcium is coming and binding with troponin C, what happens? This line, if you see this line, this tropomyosin, it will move forward. It will exhibit this actin sites or myosin binding sites to surface, to environment. What happens? This line shifted to right side or left side. This line will be shifted. Then there will be exposure of sites. This exposure of site, scientifically in book they mentioned unmasking. in book scientifically they mention unmasking of the myosin binding sites will be happen whenever the calcium which means whenever the initiation or contraction is going to happen, then what happens, unmasking will happen. If the muscle is at resting condition, what happens, masking, right? Opposite, right? Whenever there is muscle is in rest condition, what happens, masking will happen. Whenever there is an activation of muscle or contraction has to take place, then what happens, unmasking. Now what happens, this unmasking is happening. Now to these sites what will come, myosin will come. Now we will discuss how the myosin is look like. It is like snake. This is one. So I will draw with another color. This is called one myosin filament. If you see, this is one myosin filament. This myosin filament is made up of what, myofibrils, or this word, there is one word called monomers. So this is one monomer, this is another monomer. So if you see, there are two portions. One is what, tail portion is there, another one is head portion. So this is what, head, and here there is a little bit called neck portion is there, and entire thing is called as what, tail portion is there, right? So initial they segregated this entire this myosin filament into two parts. One is light meromyosins having only tail portion and having also very light portion, and another portion which constitute head plus a little bit of neck also, head plus neck. So this portion is called as what, heavy meromyosin. So this entire myofibril is divided into two categories. One is light meromyosin and another one is heavy meromyosin. Light meromyosin's only tail region is there. Heavy meromyosin head and neck portion is there. A little bit of head and neck portion. Little bit of head and neck portion is there. So in this head, what are there? That is very important. In this head, there are two regions. One is responsible for ATP binding site. One is called ATP binding site. Another one is called actin, not active, actin binding site. Initially we have actin molecule is there, and here also there is one site. I said this is called as what, myosin binding site, and also said the same name is also called as what, active, not actin. This is actin, but it is not having actin binding site. It is having what, active binding site. This is myosin, this is having what, actin binding site. This portion, this is called as what, actin binding site. This will go and bind with that, and this portion is responsible for the ATP binding site for the ATP hydrolysis. Understand? So this was the clear structure of actin and myosin. Whenever the contraction happens, this will be done. Tomorrow class we will see how the contraction means, how the attachment of these two will be happen. Tomorrow we will see, right? So understand the structure and function of skeletal muscle, which is the sarcomere part. You have to understand the diagram and what happens when contraction will happen. What are the bands or what are the zones that are going to increase or decrease or remain same. That is the very important. After that, coming to the structure of myosin filament and also the structure of actin filament, you have to remember the structures. Tomorrow class we will discuss the sliding filament theory. Understand? Today we will wind this topic, and if you have any doubts, doubts regarding topic and regarding this fees details, please contact the number which we given in our shorts, go for the shorts, you have all the contact access. Thank you for listening. అర్థమైందా నాన్న స్టూడెంట్? ఓకే రైట్, వాళ్ళు మా తాత వాళ్ళు పడుకోండి. తిన్నారు కదా మీరు?